Hello and welcome to part 2 of chapter 15. Here we're going to start to get into eukaryotic gene regulation in more detail. One of the things we see with eukaryotes is a lot more of these sort of positive and negative influences on whether a gene is transcribed. Now we saw that with prokaryotes and their operon system, but it was a little more straightforward where one repressor or one activator. In eukaryotes, there may be many transcription factors, and that's we see in this little cartoon here, these upstream elements where these proteins are binding to the DNA at different places, and some are having a negative influence, some are having a positive influence, some interact with each other. So there's a lot more going on, and you can almost think of it, a lot more fine-tuning is happening with expression of eukaryotic genes. Okay, so here's a question for you. The cells of your pancreas differ from cells in your diaphragm because why? All right, yeah, so they express different genes. That's really the crux of it. They don't contain different genes. If we think about eukaryotes in general, any individual organism, most of the cells are genetically identical. Exceptions would be gametes, for example, and there are some other exceptions, but those are in the minority. Most of your cells are the same. It's what genes are transcribed that makes them different. This cell is going to be a liver cell. It's going to produce those genes that are important for being a liver cell and functioning that way, or a cardiac muscle cell. Those are a different set of genes, but genetically those cells are still the same. And so we've talked about this idea before that gene expression is regulated at many different levels. And so we see this here in this cartoon where all these different places we can regulate gene expression, and we'll go through each one of these. And this other idea that in eukaryotes, especially multicellular organisms, you have this regulation for specialization. So this is a liver cell, this is a skin cell. These cells have very different functions. And so here's the places where we can regulate gene expression just generally. So even at the DNA level, regulating chromatin structure can turn genes on or off. At the transcriptional level, should we even transcribe this gene to begin with, activators, repressors, the right transcription factors, how they interact with each other that you've already heard about. And then after transcription, the stability, the lifespan of that piece of messenger RNA. There's other little bits of RNA that can actually bind to messenger RNA that block its translation. And once you even have a protein, the protein is activated or inactivated, it's modified, ultimately could be broken down and destroyed. All of those are ways that gene expression can be controlled. So let's start with chromatin structure. Now remember this slide, you've actually seen this a few times. So in a cell, normally, you've got the DNA, it's wrapped around histone proteins. That's how it's packaged when it's doing its general functioning, not dividing. You have heterochromatin and euchromatin, so the more densely and less densely packed chromatin. And again, if we look at how it's packaged around these histones and whether it's more tightly packaged or not, that's going to influence whether the machinery that can transcribe a gene can actually get there. So if your chromatin is really densely packed, RNA polymerase, how can it even get near the promoter, right? And so DNA needs to be unpacked or unwound a little bit from its more condensed form to get at those genes. And so that can be a regulation mechanism. So a few things can happen. So something called histone acetylation. So that adds acetyl groups to histone proteins. That will loosen the chromatin structure, kind of unwind it, open it up. So if it's a region that needs to be transcribed, that could influence whether that's transcribed or not. It'll loosen it up. Now the machinery, RNA polymerase, can get in there. DNA methylation. So this is a direct modification of DNA. Here we see the addition of methyl groups to certain residues. Here, like a cytosine gets an extra methyl group. Where methylation occurs, we see decreased transcription of those genes. So again, at the DNA level now, before transcription's even happening, we've either, shut it down. We could by deacetylation, you take those acetyl groups off, DNA will pack more tightly. Acetylate them, it'll unwind and open up. Methylation, sort of the opposite. Methylating will lower transcription. Demethylation will increase transcription. And so we can see the example a little more closely with the DNA methylation here, specifically in the promoter of this particular gene, where you have this normal, typical cytosine. This will be transcribed. Where you have the methylated promoter, you'll see reduced transcription. And here we see kind of highlighted here, these cytosine residues have that methyl group attached to them. And so that's going to decrease transcription. Now you might imagine, well, okay, that's great. But as soon as that DNA strand is copied, you're diluting that methylation. So that can happen as we see here. So think semi-conservative replication. Here's our new daughter strands. Only the parent strands are methylated here. But if that opposite strand's methylated, there's mechanisms that cells have to reproduce that methylation pattern, like we see here, this fully methylated DNA. Now, this also introduces another really interesting thing, that methylation is something that can occur during an organism's lifespan. So there's the underlying genetics that you get from your parents, but then methylation may occur for different reasons in different genes and regions throughout an individual's lifespan. And that can influence the expression of those genes, and those methylation patterns can also be passed on to the next generation. So that's also really interesting. And this is called epigenetic inheritance. Epi means upon, and so this is like saying you have the underlying genetic information, but this is built upon that. So what happens during an individual's lifetime, some of these methylation patterns that can occur, those can be inherited. And so it's not just the underlying genetics alone, but the experiences that individual has if they influence some of these methylation patterns. And so some examples here where we see epigenetic modulation, diet, toxins, medicine, psychological, all these things are potentially heritable, where as before we really understood about epigenetic inheritance, it was all about, well, what's the underlying genetics? If you have this gene, that's that, and so forth. But we now know that it's a little more nuanced than that. I will say that the underlying genetics are still the biggest part of inheritance, but you can have these subtle differences as a consequence of epigenetic inheritance as well. All right, so thinking about chromatin modulation, which of the statements here describes the role of histone acetylation, DNA methylation in gene regulation? Which one is correct? Right, so methylation is going to decrease transcription. Histone acetylation, that's going to open up that chromatin and increase it. Now, a bit of a callback to an earlier chapter where we looked at regulation of an entire chromosome through X chromosome inactivation. That's definitely going to influence gene expression when an entire chromosome is inactivated. And so you might remember this information where we see at some point in development, one X chromosome in a given cell is inactivated in mammals. And so we saw that example here with the cat, where the coloration for black or orange is on the X chromosome, where we see the black allele inactivated. That's where you'll see orange patches of fur, and then vice versa, where the orange is inactivated, you see black patches of fur. And so these condensed chromosomes are called bar bodies. And so how does, think about how that happens. The whole chromosome becomes inactivated, and then any of those genes, this is fur color, a big deal, but any of the alleles on, say, this version of the X chromosome, those are done in those cells. They are inactivated completely. So that will influence gene expression. And we saw this before. How does that work exactly? You have the production of these transcripts, this zist RNA that will bind to the X chromosome and it basically encourage it to condense super, super tight, and none of the replication machinery can now get there. All right, so let's come back to transcription now. So we've talked about the DNA level. Let's think about transcription. So we got our histone acetylation, our chromatin unwinds, things are opening up. Like, all right, this gene can be transcribed. So that's step one. That's really important. What is involved next for this to occur? So what's going to happen? Well, it helps to remind ourselves about the structure of a eukaryotic gene. So we have a promoter, like we see with prokaryotes as well. You got introns and exons. And then further upstream of the promoter, though, you have what are called proximal control elements. Proximate means close. And then further upstream, sometimes very far, actually sometimes even in introns, there are enhancer regions. And so this is distal, meaning further away, these distal control elements. And these go back to that figure where I showed you all these potential transcription factors interacting, having a positive influence, negative influence. That's where these will bind to these control regions. But let's look at the promoter first. You always need these general transcription factors to bind to the promoter. Now, this is of any gene, so that's why they're called general transcription factors. They'll bind to that promoter for any gene. So here's a promoter, and the transcription factors are more or less the same for any particular gene. And that allows for a site, or marks that site for RNA polymerase to bind and for transcription to occur. And just having these, though, isn't usually enough. So you got our general transcription factors, RNA polymerase 2, great, but that's not usually enough to get things going. You need that gas as well. So you need to put ore on the gas. Where does that come from? It comes from these other transcription factors. And so these are called specific transcription factors. So these are going to be specific for a particular control region, like this particular enhancer that happens to be up from this gene. And they'll influence very specifically whether that gene is switched on, or maybe it has a negative effect, could be a negative regulator here as well. And so these are really the crucial determining factors whether certain gene is switched on or not. Here we can see this in a little more detail. So we've got our general transcription factors binding. Great. Those are necessary. We have these mediator proteins, this DNA bending protein that brings these activators in close proximity with the actual promoter. And it's not until all that comes together that we finally get that transcription. And just again, a side note, not all of these transcription factors are activators. Some of them can be repressors too. And so here's that figure again showing positive influence, negative influence, transcription factors interacting with one another. So you can imagine here, like in this last bullet, when you have many control regions, so say a gene has a dozen control regions, that's still a lot. There's so many possible combinations about whether this one's having a positive influence, does it interfere with this one, or does it activate the other one? You can start to see the scenarios get complicated. But remember, this is all about fine-tuning as well. Keep in mind, thinking about what makes the liver cell different from, say, the lens cell of an eye. The genes are all the same. General transcription factors would all be the same. What's going to make the difference between whether a certain gene switched on or off are those specific transcription factors. And say in this liver cell here, we have this, let's see, this little gray transcription factor. It is found in both the lens cell and the liver cell. So those may be important for transcription of this kind of gene, but it's sort of semi-specific. In the liver cell, though, we have this red one and this yellow one. And when they're all present, this albumin gene can be expressed. And notice down here this crystalline gene in the liver cell, that it's pointless. There's no need for this at all. This one transcription factor binds, but the other ones aren't present, so this is never transcribed. Take the lens cell of your eye. Again, the little gray transcription factor binds to this albumin gene, but the red and yellow ones, they're not here. So it's not going to work. This crystalline gene, of course, though, in this case, we have the orange and the pink transcription factor. Those are present. That's going to switch on this crystalline gene, and that's what's needed in the eye. You don't need albumin being produced in the eye. All right, with that in mind, check out this question and pick the correct answer. All right, yeah, it's A here. We can rule out some of these other ones. C, for example, it says operons. We don't see operons in eukaryotes. That's only in prokaryotes. B here, it says these genes share a single common enhancer. No, it's in fact, each gene, you might have multiple different enhancer regions. So that one doesn't work either. All right, so we've got our transcript made. What happens next? So just transcribing our mRNA, that doesn't constitute gene expression. There's other things that need to happen. So let me give you an example as a question. So a specific gene codes for 3 different but related proteins. So this is a specific gene, 3 different proteins. What could be the cause of that? All right, this is something you've seen before, this idea of alternative RNA splicing. So we have this initially this gene and the same transcript that's produced from this gene, but by splicing it in different ways, we're going to produce 2 different messages. They're similar in some ways, but also different here. This one has exon 3, this one has exon 4. And we could look at this as another real world example. This insulin receptor in, say, liver tissue. And so in liver tissue, we have exon 11 here present, and we end up with an insulin receptor that has low affinity for insulin, whereas in skeletal muscle, this exon actually gets cut out, and we end up with this transcript lacking that exon 11, and you end up with a protein that's an insulin receptor that has high affinity for insulin. So just from the same gene, we've produced 2 different possible outcomes. And remember this one, this is so important when we think about genetic diversity. By the time we get to transcriptome diversity, because of alternate splicing and some other modifications, we have more possible transcripts than there were genes. And then we can produce all these different proteins, which those can also be modified and so on. And so this is one example, RNA splicing, differential RNA splicing, is one example of post-transcriptional regulation. Another couple of examples here are RNA degradation and RNA interference. And so, degradation is just as it sounds. The RNA transcript will break down over time. And, RNA transcripts only have a certain lifespan. There are enzymes that will eat away at the ends of this molecule. What protects that to some extent? We've already talked about this before, this poly A tail. So it takes a little while for that tail to get chewed away at before we start to get into the actual transcript here. That 5' cap, important for initiating translation, also protects this end of the transcript. And with these modifications, that means eukaryotic RNA, which has these, it tends to be more long-lived than prokaryotic RNA, which does not have these modifications. Another really interesting thing we see is tiny little pieces of RNA acting as regulators of messenger RNA. And it's interesting because not that long ago, maybe 20 years, maybe 25 at this point, there were people studying RNA. They would separate out the RNA from a cell culture, for example, and they would find a lot of little bitties of RNA. And for the most part, people thought, well, that's just degraded RNA. That's just garbage. We're really interested in the big stuff, the transcripts. This little stuff is just junk. Now, depending on the experiment, some of that could have been degraded RNA, but we've learned since that there's a lot of tiny bits of RNA that are super important, and they're critical regulatory molecules for gene expression. And these are called, one class of them is called microRNAs. So they're only about 20 nucleotides long, which is why, people sort of thought they were just junk because they're so tiny. And they can be complementary to a piece of messenger RNA. And so what happens, you've got these single-stranded molecules, and you've got this little tiny matching piece. It will bind, complementary base pairing will happen. And now you've got a short stretch of your message that's double-stranded. And so this can block translation. It just gets in the way. It can also act as kind of a recognition site for machinery that breaks down double-stranded RNA. So, there's a double-stranded piece of RNA. Let's start chopping that up so that it can enhance the degradation of that piece of message. Another group of these small RNAs are called small interfering RNAs, and they function very similarly to the microRNAs, but they're produced in a slightly different way. Again, for the purposes of a Gen Bio class, know that there are these different kinds, and they do end up having similar functions. And let's see how one of these functions works. So we talked about they can just block and get in the way of your translation machinery. But here we have this is our microRNA in this example, but we'd see something similar with the siRNA. It's actually associated with this enzyme. It's called RISC. And what this does is when our little piece of microRNA binds complementarily to our piece of message in this case, that this enzyme complex CLIP will cut it in half. And this will basically has destroyed this message, right? If this is in the middle of a gene, that has destroyed the message. All right. So since Watson and Crick described the structure of DNA, which of the following statements explains why the function of these small RNAs is still being explained? All right, so the bottom line here is we're still learning about this stuff. So this is relatively new in the last few decades of even understanding that these are there. And so we still have a lot to learn, but there are good chunks of our genome that produce these small RNAs, and these do have a very important role in gene regulation. Something else we see with RNA is something called RNA editing. This is another area that's we're still learning a lot about, but let me give you an example here. So tissue-specific RNA editing. So this is a certain protein, the mRNA for the protein in the liver here, and it produces this amino acid sequence, and it's going to produce a protein or polypeptide that's over 4,000 amino acids long. In the intestine, this also produces this transcript, but an edit takes place. This is after the RNA is produced. It converts this C to a uracil, and this introduces a stop codon. So this isn't a mutation at the sense of the DNA level, but it's edited RNA. It still produces a functional protein that is about half the size of what you see in the liver, and this protein has a different function. All right, so what happens after we do produce a protein, whether it's in this case or any other case? You've also seen this slide before. Proteins might need to be modified, combined with other polypeptides, cleaved, have other groups attached to them. All kinds of things can happen to ultimately end up with that target protein. And then we see this slide yet again, where a reminder, this idea that we have genomic diversity, and from that, our protein diversity can be so much greater because of the variations that we can insert at these different stages. So that's one thing that can happen, post-translational modification of proteins. what about you just don't need that protein anymore? I mean, proteins will break down over time, but they can also be targeted for breakdown when, again, we don't need that one anymore. That function isn't needed. We can switch it off or, in this case, destroy it. And there are these other proteins called ubiquitin in our cells. And proteins that are to be degraded are marked, if you will, with ubiquitin. So these ubiquitin proteins are added by an enzyme to those proteins marked for destruction. You can see here. Ubiquitin's being added. You need a few of them to be added for this thing to get marked for destruction. And once they are on there, that marked protein will be sent into this structure called a proteasome. This is also made out of protein. And I almost think of it like a garbage disposal. It will take our marked proteins and just grind them up into pieces. And so here we have our polypeptide fragments, the little bitties from our protein. Those can be broken down further. Amino acids can be recycled, and our ubiquitins can be recycled too. And so here's our ubiquitin ligase enzyme that now can tag the next protein that is marked for destruction. And so we've seen from the chromatin level, modifying DNA or histones, packaging, unpackaging chromatin, all the way to the point of having produced a protein and modifying that, or ultimately maybe degrading it, how eukaryotes can regulate gene expression.